This protocol shows that radiation influences extracellular matrix properties of adipose tissue in a murine mammary fat pad. The addition of radiation in a decellularization model has not been done before. This technique uses several washing steps that each serve a unique chemical purpose in the decellularization process. The specificity of this technique allows for gentler chemical washes to better preserve tissue properties.
Breast cancer relapse occurs following therapy, especially in triple-negative cases. Evaluating how the irradiated extracellular matrix alters tumor cell behavior will lead to important discoveries about recurrence mechanisms.
After irradiating samples using a cesium source, transfer the irradiated MFPs and complete RPMI media into a biosafety cabinet. Fill 6 cm or 10 cm dishes with enough media to submerge the MFPs. Incubate at 37 degrees Celsius with 5% carbon dioxide for 2 days. First, place the MFPs in 6 cm dishes with 5 ml of trypsin EDTA solution. Spray and wipe the dishes with 70% ethanol and incubate at 37 degrees Celsius for 1 hour. Use 0.7 mm strainers to wash the MFPs with deionized water by pouring water over the tissue 3 times. Use forceps to manually massage the tissue in between washes. Next, briefly dry the tissue on a delicate task wipe and weigh it. Place the dried tissues in a pre-autoclaved beaker containing an appropriate sized stir bar, and cover the tissue.